Hi, everyone, and welcome to a very special video. We made this announcement recently that Dr. Travis Stiles is joining the MStranslate team, and it's my pleasure to welcome Travis here for this video today to do our first piece of content together. Travis, welcome. I guess you've been on MStranslate a few times before, but for people who may be new to you, do you just want to give us a really quick introduction to who you are and the work that you're doing, which is going to tie really nicely into what we're talking about today in terms of remyelination?
Sure. So Travis Stiles, I'm living in San Diego, California, lovely San Diego. And I'm the CEO and co-founder of Novoron Bioscience. Novoron is developing non-antibody-based biologic therapies to promote regeneration of tissue in the central nervous system. Our two main programs are in spinal cord injury and, of course, MS. So we're developing therapies to promote remyelination in multiple sclerosis. Yeah. So I don't know what else to say. Is that good? I don't know what else to say.
No, that's great. So, I mean, we'll be talking a lot more about the work that you're doing in future videos as well. But ties in really nicely because today we're going to be talking about some of the research that you and I heard at ECTRIMS on remyelination and myelin repair. And we've asked some of our community members to submit questions in advance of this video. And nicely, they've asked questions which tie in really nicely with some of the presentations that we heard. So we can just fit it together by answering those questions. So first question that was submitted by Pam was around whether or not age or EDSS impacts on remyelination therapies or the ability for myelin repair. And I guess this ties in with two presentations that we heard, one about bexarotene and one about oportunumab, which both touched on those. Do you want to start us off sharing some of your thoughts on either of those presentations?
Yeah, well, so I would say at a baseline, when you're talking about age-related response, we already know that the remyelination capacity in the brain and spinal cord is going to be better in younger people. Same with kind of neuroprotective or neuroregenerative kind of responses. So I think that there's always going to be a baseline assumption that younger patients will respond better to certain things. Where that kind of gets turned on its head, however, is in the Optic trial, it actually appeared that the opposite was true, that the older patient population actually had a stronger response to treatment than the younger. Now, I think that there's a clear caveat there that none of the results were statistically significant. But I do think that the results were, you wouldn't look at them and say, oh, there's nothing there. I mean, there's the trial that was presented was called the Affinity trial. It was actually an extension of a previous clinical trial they had done called the Synergy trial, which was a sister trial to another trial for acute optic neuritis called the Renew trial. And there's always been reason to believe that there was something going on, that there was an effect that was observable, but they couldn't really. The reason they went to the Affinity trial was to try to find a way to tease out what effect was there. So trying to get back on topic, that is an actually very interesting finding that there might be a population that's actually older that responded well. In the other one, the RXR agonist.
Sorry, just to jump in before we jump onto that one, just to keep going with the oportunumab trial. So people may have heard of, I guess, this before in terms of anti-LINGO may be the term that they're aware of and have heard about some of these previous trials.
That's a good point, yeah.
But in terms of, so you mentioned that trial wasn't positive in terms of the outcomes, that it was essentially didn't meet what it was hoping to achieve. But talking a little bit before this and during the conference as well, I guess we have some thoughts around maybe why that is in terms of the group of people that they were looking at and why it might make it difficult to achieve a positive response, but also potentially why they did see a better response in older individuals. Do you want to talk about that just quickly?
Yeah. So, I mean, I've been familiar with this set of clinical trials around oportunumab, the anti-LINGO monoclonal antibody therapy, for quite a while. I think a lot of people were very excited. The first trial results that came out were the Renew results. So Renew was the optic neuritis trial. And when those results came out and they actually hit their endpoints, it was very exciting. It was, in practice, what that meant was it was really the first clinical proof of concept that you could promote remyelination in a human patient. And that was, that's exciting, independent of like if a drug gets approved or not. And so there was a lot of enthusiasm behind the Synergy trial, which was in a relapsing remitting MS population. When the Synergy trial didn't hit its endpoints, there was a lot of kind of consternation and confusion because the Renew trial had been successful. And one of the things that I think from the beginning I was frankly a bit frustrated with is the trial design was very different. In the acute optic neuritis trial, literally the patient inclusion criteria was new lesions. So in what that means, when you have somebody who has a new lesion, they're having active disease. And the reason I think that is very important is because opicinumab is a monoclonal antibody. Antibodies are big molecules that don't get in the brain, at least not well, under normal conditions. But if you have an active lesion, kind of by definition, the blood-brain barrier around that lesion site is compromised. So if you were ever going to put an antibody into the bloodstream and have it get into the brain, it would be when there's an active lesion. And not only would it potentially get into the brain, but it would get into the brain right where you want it to go. The thing that frustrated me about Synergy is that they didn't stratify based on that. They actually had very kind of what I thought were vague inclusion criteria. But when the kind of post-mortem analysis of that failed trial came out, there was a lot of, I guess, theorizing that there was a subpopulation in that trial that they thought had actually responded. And you obviously can't do a trial that way. You can't say, okay, well, we didn't see what we wanted to see, but if you squint and look over here, it looks like there was a thing. You can say that, but you then have to build on that. You have to take that, whatever you thought you saw, and then you have to make it its own thing and prove that what you think you saw, you're actually seeing. And so in theory, there's been talk for years, I mean, probably since 2017, 18, that they were going to try to expand on this supposed like subpopulation and try to build on that. So there's been a lot of, I actually didn't really know where that trial was at until they presented this data at the conference. So I was very interested to see what the results of these newfound efforts to kind of revitalize this approach was going to bring. What was disappointing is that it doesn't appear that the stratification or the way that they tried to expand into this new trial addressed what I feared was the limitations from the first trial. Again, so if you really were to try to look at the broadest terms, why you might see a benefit in the optic neuritis trial, not the Synergy, it's disease. It would be disease state. It seems like an obvious point where you could say this is why this is different. And so you would hope that if they saw something in the Synergy trial that would give them an idea that there might be a better place to look, it would have been that. But they actually stratified against active disease. I think it was 24 months. You couldn't have had an active lesion or a new lesion form for at least 24 months. And I might be wrong on that, but regardless, there was actually a criteria that you couldn't have active disease and you couldn't have had active disease for a very long time. So if the hypothesis is that you need to be able to have a compromised blood-brain barrier for the drug to get to the area where it's needed, they actively chose against that type of patient profile. Now, the reason I give you all that background and kind of meander a bit in my explanation is because when you start to talk about, well, what if this age-related phenomenon is real? What if it really is that older patients did respond and younger didn't? Well, if we stay on the kind of mindset of maybe the blood-brain barrier is playing a role in some of this, we know that the blood-brain barrier integrity goes down with age. And so it's possible that part of the reason that they saw benefits, like, this kind of like counterintuitive improved responsiveness in older patients goes back to the same basic assumption we had in active versus remission disease, that the patients just had more bioavailability to the brain because they just by nature of being older have a more permeable blood-brain barrier.
Yeah. So yeah, I mean, it's one of the things, and it came up a lot during the conference, around a number of different things is making sure that if we're trying new treatments, are these treatments actually being able to get to where they need to go? And so I guess that's still a question even in this failed trial that we have, as you say, may play. If your thoughts on that are true, may explain why the older people showed maybe a slightly better response. Now, we talked about on the flip side, we've got bexarotene, which is a trial coming out of the University of Cambridge. Now, this, on the other hand, was a positive trial. They did show that bexarotene was able to promote myelin repair. Now, it's not going to be pursued, as we've talked about a few times on MStranslate already, because of the sort of severe side effects that it comes with that were far too unpleasant for it to be continued as a treatment. But they also, in this follow-up study that they presented this year, talked about some age-related differences that they saw, shifting more towards younger patients, maybe having a better response. So how do we, I guess just as a general sort of thought on all of this, how do we sit in terms of what we think the role age may play? Do you think we have enough information at the moment to say younger is better, old won't? You know, how can we?
I think it's going to be mechanism specific, right? Like, so there's going to be drugs that in an ideal world won't need, they won't need to be active transport into the brain. They'll have passive diffusion. There's going to be drugs that, like, in theory, there might be a peripheral effect that has a central nervous system benefit. So, I think it's going to depend on the mechanism. I think if you're going to have a drug, as any monoclonal antibody would be, that relies upon some form of friendly passive entrance into the brain, I think that there's a reason to think that older, sicker patients, which is what we saw in the Affinity trial, older, sicker patients are going to have a better response. But I don't think that we should necessarily celebrate that. I think it's good to know that maybe later in disease course and age, that there's things that might be options that wouldn't be otherwise. But they're options because the disease has gotten far enough where you're already in a compromised position. And the goal really should be that having a compromised blood-brain barrier is not a positive thing in general, right? So I think that from a dogmatic standpoint about how we should view this, I think the fundamental reality is there are a few things in the human body in terms of disease responsiveness, healing response, anything where age isn't a factor. Everything about the human body says that, at some point you get old and stuff stops working as well. So I think that in general, if we find a good drug that does a good job and it has good kinetic properties and good bioavailability, you're probably always going to potentially see a stronger response in younger patients. And that might be difficult to tease out, because if you're controlling for age, they're also going to have a stronger baseline response. So in theory, the relative efficacy should still be a difference you can observe, right? So if you're comparing 30-year-olds to 30-year-olds, it's probably not going to be a striking difference if you're comparing 60-year-olds to 60-year-olds. Mm-hmm. But in practice, if you start, if a drug gets approved and you're monitoring patients on like a post-marketing trial and you're just trying to see what happens, it's probably completely reasonable that if you follow the 67-year-old patient cohort, that the disease progression benefits, the efficacy is probably going to be lesser than the younger patients. And that's just probably a reality of where we're at. I also think that my fundamental fear is, like, if we have a result like this, where the thing that makes the pieces that are otherwise potentially a logical fit is, it's blood-brain barrier permeability, it's compromised blood-brain barrier, then I think that there's another opportunity here to look at, okay, is there ways we can do this that doesn't rely on passive breakdown of the blood-brain barrier? Is there a modality of treatment? Is there a way that we can, because this is exciting, right? Like, if you can find a way to make the myelin sheath repaired in the brain, that has an amazing potential benefit beyond just MS. So, like, if antibodies aren't the best way to do it, is there a better way? To me, those are the questions that sometimes don't get asked in large bureaucratic companies and big pharma, that I think are worth thinking about.
Yeah, and that's something that we've talked about before. And if you watch Travis's talk in New Zealand from 2016, I think it was, that we were there, it's something that you talk about. It's probably a topic for a whole nother video where we talk about that sort of approach. But so I think, I guess our a takeaway message from all of that is we don't know yet, in terms of there's nothing to suggest that people of a certain age won't respond to myelin repair therapies. It's going to depend on whatever the therapy is. Younger may be better, and there are obviously ongoing trials that are looking at compounds that maybe can slow that aging process, and this is talking about metformin, and maybe we'll do a completely separate video on that as well, because I think we both agree that that's a really interesting thing, and we know that our community is interested in it as well. So that's where
I definitely think that could be an entire video, because in theory, if that metformin combinatorial therapy works, that opens a whole Pandora's box, a beautiful high potential Pandora's box of how we view multiple sclerosis and how we treat things in the future. And not just in regards to how we use some things like metformin. I think it can be paradigm changing in how we view a lot of the ways that we treat multiple sclerosis patients.
Yeah. And so I guess it's a good point to say in this, that these videos largely are going to be Travis and I getting to share our opinions on things. We're going to take a little bit of freedom in this, in terms of, we'll obviously be talking about data and using the science to guide us, but you will be getting to hear our thoughts on it, which is different, say, to some of the summary videos that I do, where I really just report back on what's been shown. So if you have a topic that you want to hear us talk about and hear our thoughts on, share it in the comments below, because we're keen to be doing these. We enjoy getting the opportunity to just have a chat and talk about these different topics. So I guess
and Brett is an excellent scientific communicator. He does a really good job. You do a really good job of breaking down the complexity of scenarios and doing a good job of communicating it in a digestible format to your audience. I love when we get a chance to sit down and do this, though, because there's so much, especially with how prominent social media is. A lot of the public rhetoric around kind of scientific topics, it gets ran around in the ringer among bubbles of people talking about stuff. Whereas I think it's helpful when you hear two scientists actually expressing their opinions and going back and forth on stuff, to see what that debate could look like. Instead of just seeing what the guy on the news tells you, or what the paper tells you, or what your neighbor who doesn't know much about science tells you, to see an actual debate and a conversation, not just a lecture or a press release. Because I do think that there's nuance that can spark interesting thoughts, but I also think it's really important for the community and for advocates to know how we think about the problem, not just how we digest and convey the new findings. Does that make sense?
Yeah, definitely. And I think I think what is going to be really exciting for our community is having you involved in these discussions, who's someone who is on the ground still doing all of this work and in this space and going through all of these problems yourself and having to work through all of these. It gives a different perspective to me, who's now, I guess, outside of the lab, but getting a broad sort of overview of what's going on. So I get that perspective and get to see lots of things, and you're in there doing it. So I think that's going to be really exciting. The debate concept is interesting. If you want to see Travis and I do a debate on a topic, give us a topic and maybe we'll choose a for and against for that, and have a bit of an argument. I mean, a friendly
even
debate.
If we agree, force one of us to disagree, that'd be fun.
Yeah, we'll look into that. Next question, I knew we said we'd keep this short, and we're probably already over time, and we've got many more things to talk about. Debbie asked a question around whether or not myelin repair is possible for secondary progressive MS. Let's broaden that slightly and talk about progressive MS in general. So I guess some quick general thoughts on that, and this related to a talk again at ECTRIMS for the Abutolast trial, which we have a few little thoughts on as well. So do you want to start us off on that?
Yeah. So I agree. So in general, like, when you think about progressive MS, I think the fear that people might have is like, if you never get to remission, can you repair? I actually think that might be the wrong way to look at it. Like I already said, I actually think during active disease, or maybe in the weaning hours of the most recent attack kind of backing off, I think is the prime time to treat. We have a lot of great disease modifying therapies. We have a lot of drugs that can go out there and it can affect the immune response. But let's be honest, like, the holy grail right now is to be able to take the damage that's already occurred and reverse it. And so I don't think, in my personal opinion, I actually think that, if we're really being reductionist about this, a progressive disease state is just a persistent active state. So in theory, in my personal opinion, I actually think that absolutely there's an opportunity to promote remyelination in that active disease state. Now, maybe it might be difficult to see because you might be having remyelination and demyelination happening at the same time, but the nuance there might make it difficult to observe the result. But I actually think that might be the prime time to go in there and have an effect. And I think that one of the things that you'll often hear people talk about as a theory, it's not something that you'll hear as like a prominent hypothesis people put out there, is I think that in some ways, if you think about secondary, SPMS as basically a kind of the end result of a worsening RRMS, it's basically the body has just kind of lost the ability to have these breaks and attacks, right? There's reason to think that if you were able to kind of promote a remyelination response, you might actually be able to tip the scales back from an SPMS back into more of a relapsing remitting type state. And theoretically, you might even be able to do the same to a primary progressive, where maybe if you give the body a little bit better chance to recover, maybe you give it the ability to stave off the immune attack long enough to go into remission. But I think that there's also a lot more complexity in terms of progressive MS that might not account for. But yeah, I mean, in general, I think secondary progressive, especially, I think there is absolutely room to think that there's remyelination capabilities there. Frankly, like I think what the anti-LINGO opicinumab trial might have taught me or made me feel even stronger about is if you've had a lesion for 2 years and that's your disability, none of the approaches that we're currently looking at right now are probably going to do much to reverse that, because you're looking at things that are beyond disease pathology. You're looking at long-term fibrotic changes to focal regions of brain tissue. And that's frankly not unique to multiple sclerosis. There may need to be things that we do or think about that aspect of brain pathology in order to kind of reset that tissue to make it even be receptive to remyelinating therapy.
So can I just stop you for a second? Can you explain that? Can you simplify that a little bit more in terms of what you're talking there about the damage that we're looking at? So
yeah,
it's more that you're talking about axonal loss, that you're going to end up losing nerves. Can you explain to people?
So I mean, there's a lot of pathological hallmarks there. Obviously, you get actual axonal loss. You might even get necrosis, necrotic tissue. You get kind of a vacating of productive glial cells, and you'll get kind of an influx of inflammatory cells and fibrotic cells, fibroblasts, things that come in and just lay down a crap ton of matrix proteins. But in general, if I really wanted to simplify it, when you get damage to the central nervous system, in the acute phase, you have the disrupted myelin, and in the longer term phase, you get glial scarring or gliosis. And the terminology about how we refer to that, like whether you just referred it to as a gliotic response or gliosis or scarring, they're all different kind of, it all kind of circles around this generic idea of fibrosis. You get a remodeling of the microenvironment to be, to have a lot of extracellular matrix deposition. And the way I think it's a good way to think about it is like, if there is let's say your house gets hit by a comet, and ideally, what the best way to recover from that is to bring in a crew, take the broken pieces away, haul them out, get all the edges cleaned up, and then bring in a construction crew to build up the damage that was lost. The problem is that because the blood-brain barrier can be so important, and because there's such a rush to try to literally sometimes stop the bleeding, instead of having this patient response to come in and clean up the mess, get everybody out of there, take the demolition crew and the cleanup crew, have them do their job, take them out, and then bring in the construction crew to rebuild, there's this rush to just fill in the hole. And so it's almost like they just bring in a cement truck and fill in the crater. It's very difficult to build a house where a bunch of cement is. And it's that component that I think becomes problematic. Like in theory, if you really did need to stop, I say stop the bleeding because it does have some relevance here in terms of the blood-brain barrier breakdown, the influx of blood-borne cells. There actually is some bleeding component there. But in general, it's just to patch the barrier again so that you stop letting these immune cells in. So it really is almost like this pouring concrete in the hole. Now, in the real life example, what you would then need to do is go in there with a jackhammer. Once everything's done, all the comments are gone, whatever aliens, you've taken care of it all. You come in with a jackhammer, you break up the cement, you get it all out of there, and then you can still rebuild. And I really think that's an apt analogy where there's still the potential to rebuild, but the fibrotic response is a hindrance. And that fibrotic response is something I'm Novoron's uniquely interested in because we think it's important. But I just think that when you're talking about something like the Optic trial, where they're literally talking about the majority of these patients not having a new lesion for 2 years, there's a big wall to climb in terms of how you're going to go in there and break up that concrete with jackhammer for 2-year-old concrete slabs, right?
Yeah, I think we need to keep in mind here in terms of we're not necessarily going to be able to repair all of the damage that's there. We want to, in a perfect scenario, as new damage occurs, and hopefully with some of the existing therapies that are there to prevent new damage, that's not happening very much. But if new damage occurs, we go in and repair that almost straight away, rather than necessarily trying to repair damage that's 20 years old. That's not going to necessarily be possible.
And conflating the two, all the trials that we just looked at, they don't differentiate. They're just talking about lesions, and they'll measure lesions with different hyperintensities and different enhancements and whatnot. But in general, they think of remyelination as remyelination, and the idea they don't really discriminate between older and newer lesions. And I think that that's a mistake. And the reason I say I think it's a mistake is because what I don't want your audience to think is I'm saying that if you've had lesions for a long time, you're out of luck. I don't believe that. It's just, and I also don't believe that it's the wrong approach to try to do just the remyelination approaches now, because you want to have to take reductionist approaches. But I think that if you're not stratifying, if you're not considering the differences in what types of lesions and the level of fibrosis in a lesion, then you're minimizing your ability to see statistical significance. Because if younger lesions might be more responsive, but you're looking at all of them together, you're going to kind of hide that response. But if we were to find a good remyelination therapy that worked on new lesions, boom, done. We've hit that threshold. Let's say it doesn't work on older lesions. Fine, we know it works. Now we can just look at the fibrosis, and then we can do combinatorial approaches to look at the fibrosis as a separate pathology that would then enable the remyelination to work. But I think that nuance of being willing to think of it as potentially needing to have multiple steps or multiple modes of intervention, I think is often lost in the conversations. Which and that's not to be critical of why that is. I get why it is, but I think it's important to note that even though I'm cynical about the way some of these studies were conducted and how some of these remyelination therapies might work with older lesions, that doesn't mean I'm cynical about our ability to treat older lesions. I just don't think we'll be able to treat them the same as we do more recently formed lesions.
Yeah. And so at ECTRIMS, we heard the talk on Abutolast, which looked specifically at brain atrophy in people with progressive MS. And they saw a positive result. They saw that they could reduce brain atrophy using Abutolast in both individuals with primary progressive and secondary progressive, potentially better in primary progressive. But I guess at this point, we're not completely sure if that's through remyelination or through some other mechanism that's stopping atrophy. But yeah, I think after that big chat around these factors, I guess our message for the community, if I'm interpreting it correctly, is that we're quite positive about the ability to do this across ages, across disease types. It may just need different approaches. We need to be really careful about how we're doing the trials, because we can't take a one-size-fits-all approach. That's the key thing that we know about multiple sclerosis. People are very different, and they're very different at lots of different levels. And so, taking in all of those little details into how we're doing the trials, the therapies that we're looking at, is going to be really important.
Yeah.
All right, last few questions. We'll try and keep these ones a bit quicker. These are sort of not ECTRIMS related questions, but they are remyelination questions. I might do the first one because it's going to be a really quick one. So Andrew asked about high dose biotin and whether it's still thought to assist in myelin repair. So this is a story that we covered previously on MStranslate that had some quite positive results from early trials. However, the later trial has shown that they haven't been able to replicate those results. They've tried a couple of times now. It doesn't seem to be effective in meeting any of their endpoints, as well as that the use of the high dose biotin was found to impact on certain medical tests and give sort of dangerous results there in terms of the results that they were giving. So it's not going to be pursued. This was published late last year to indicate that that's probably not going to continue on. So that's, yeah, a little disappointing, and that we'd seen some quite positive results from that early on. But yeah, in terms of those trials with MedDay Pharmaceuticals, they're not going to continue with that. One that I know we've talked about before, and this came up from both Christian and Sandy, was talking about some early stage research that's going on by a company called NerveGen and their compound NVG-291. Do you want to share any? I know you know a little bit about this. Do you want to share some thoughts that you have?
Yeah, so I'm actually very familiar with NerveGen. So NerveGen actually started very similar around the same time as Novoron did. I actually met one of the co-founders of the company at a conference, a spinal cord injury conference called the International Symposium on Nerve Regeneration. It's one of my favorite conferences I still like to go to. It's been a while now. COVID hasn't helped. They hold it at Asilomar up by Pebble Beach. And it's one of the coolest spinal cord conferences. They only hold it every other year. And NerveGen came out of a guy, Jerry Silver's lab out of Case Western. And Jerry's a godfather in the field of spinal cord injury research. And one of his kind of like really pioneering seminal discoveries is he really characterized the role of, I mean, I don't think he was the one who like really discovered, but he did a lot of work on the role of chondroitin sulfate proteoglycans in impairing neural repair. But he is probably the guy who pioneered the use of chondroitinase to break down the CSPGs as a means to promote nerve regeneration. So, and I think that this is actually interesting because it's getting a little bit away from the MVG drug, but when I talked about like the fibrotic responses, this is Jerry's previous work on chondroitinase is a prime example of the types of things that we can do. You know, Jerry wasn't a neuroregenerative therapy, the chondroitinase ABC that he was using, but it definitely helps to break down the glial scarring that occurs in the spinal cord injury. So the MVG drug is it's a PTP gamma or it's a PTP peptide, PTP activating peptide, I believe, that they've been working on. I want to say the first times I heard him talk about it was like 2009. And part of the reason why I think that their company's onto something is they saw in their early stages kind of something very similar to what Novoron did. Part of the reason Novoron got into the MS space is in some of our early spinal cord studies, we saw myelin changes. And that's what kind of opened that up to like us going into the MS therapy route. And NerveGen really, I think, got started the same way. In some of their early spinal cord studies, they saw myelin changes. And to me, because I obviously am a very big fan of our technologies, the fact that they saw similar things makes me think, okay, well, they might be onto something too. So I'm really interested to see what happens. You know, peptides are interesting because peptides don't have the same, in theory, it's not as hard for them to penetrate the brain as large molecules like, you know, antibodies. But that doesn't necessarily mean it's easy for them to get in either. And they're just much, much, much smaller. So the likelihood of them being able to get in is still there. So yeah, I'm really interested. Jerry really cut his teeth and made his name in the spinal cord field. But I can tell you from personal experience that the remyelination, nerve regeneration, pathology, cell signaling, all that stuff are so intertwined that it would not surprise me at all that if somebody was to be able to successfully remyelinate or regrow axons, it would have the effect in the other way as well. So So yeah, I think it's something to keep your eye on for sure.
Yeah, it's certainly a wait and see. I think we've just maybe within the last 2 weeks, they've given some updates on a very early stage phase 1 trial. So this is just healthy individuals saying that they tolerated the compound. But we don't have any more information on that yet in terms of any trials in terms of MS. But Travis and I will both be monitoring it, and we will.
And I will say, the other thing I can really relate to, and I just to prime the MStranslate community here is like, they're a small company just like ours. They've raised more money than us. They went public in Canada. I did an IPO in Canada, which when I heard about that, I was like, well, that's interesting. Is that something you can do? Should I do that? Because they raised a lot of money doing that. But when it comes to why you would talk about a phase 1 safety study and why you would tout that is it's the creation of the perception of value, but they're going to need to raise more money now to do the phase 2 trial.
Yeah.
So I can tell you as a small startup company trying to develop therapies that the press release they had on the safety trial is a huge win for them, but it doesn't, and I'm not saying this to be pessimistic, and I don't have any indication. There's no reason to think that this would bode poorly or well for their disease trial, but this is in healthy patients. It's just to see if their drug is tolerated, but it's a necessary step for them to get to the next level. And it's one of the things that investors really want to see before they feel confident investing. So I would just give it a little bit of time, because they're almost certainly going to need to raise a little bit more money before they even begin the phase 2 trial, but definitely keep your eye out to see if they register a trial, because that will be the next step, to do an interventional trial in a disease population.
Yeah, definitely. All right. And our last question came from Lindsay, who is keen to understand if research has had any focus on optic nerve myelin repair. Now, this is something that we talked about at length when we met a couple of days ago, so we'll try and keep it brief, but I know it's something that you feel quite strongly about. So I'll let you answer this one.
I don't know why that's not where everybody's starting. I don't know why. I mean, with the Renew versus Synergy trials, I really wish Biogen, when they did their Renew and Synergy trials, would have just said, bag Synergy, focus on Renew, see if we can get FDA approval for the acute optic neuritis, and then leverage that success in the smaller market to then move into the relapsing remitting. I think that there is a strong case to be made that is an elegant means to de-risk an asset, to get a clear, simple means of demonstrated efficacy before moving into the more complex pathology. And the real specific reason for this is, if you have an optic nerve lesion, you have very specific consequences. You have reduced visual evoked latencies. You have visual disturbances. There's only so many things that can happen when you have an optic nerve lesion. When you have MS, depending on where your lesion is, you can have a whole variety of issues. You could have numbness, tingling, paralysis, speech disturbances, smell weird things, mood disturbances. And so if you're taking patients that have all these different hodgepodges of disease and trying to find a number that can say sicker or better, that is a hard statistical thing to do. So I really think that there should be, I really like the idea of just sticking to not sticking to, but I really think that this is hard. Nobody's done this before. When it comes to remyelination, I don't know why more people aren't looking at the Biogen example and saying, let's not make that same mistake. Let's not just go after the holy grail on the first bite and let our eyes be bigger than our stomachs. Let's do let's try to tackle the lower bar first, and then use that success to leverage further development. Because the other thing is unless you're big pharma, if you take a big swing and you strike out, like Novoron, if we did that, we would never be able to try again, because we'd never be able to raise the money. So we would want to take the more reasonable bite first to establish success and then move forward. Because I fundamentally, one of the things that's one of my mantras when it comes to Novoron is a false negative. Failing because you took too big of a reach, or because you didn't do a good study design or whatever, is not just a practical financial failing. I think it's an ethical failing. I think there's a moral component to it also. When you just try to jump to
the last thing you want is for a potential therapy to get binned, not because it's not going to work, but because it was just the study was done poorly, and then no one's going to pursue it in future because they think, well, this has been shown not to work. And yeah, that's the worst possible outcome, I think.
And here's the thing, that when it comes to Novoron, that's what keeps me up at night. Not that the drug doesn't work, Not that what keeps me up at night is if the drug works and I botch it. That's my biggest fear. And I think that fear should motivate more people. I think when it comes to making the business decision or trying to look at like, quarterly, earnings reports and whatever, I think that people sometimes lose sight of an iterative process gives you can sometimes make you have a higher likelihood of success. Stigmatizing a drug with a failure because you took a quick and dirty shot, if it compromises your ability to take the better shot on goal, I think that you're going beyond making poor business decisions, and you're erring into an ethical issue.
Well, and coming back to the optic nerve myelin repair, it's interesting that we've shifted away from it, because I guess one of the bigger sort of screening studies that we had a number of years ago now was coming out of UCSF from Jonah Chan and Ari Green doing the work that led
brilliant dudes, by the way.
Yeah, which led to the identification of clemistine, this was all done looking at optic nerve repair. And they found that compound, and that compound is now going into the metformin trial at Cambridge to look at that combinatorial approach. But it's interesting, having had a program that's generated success looking at optic nerve repair, that then it seems to have disappeared largely from there. So I think your thoughts are really valid on that.
And I do want to say, there is a lot of anti-pharma sentiment out there, and I know that it might sound like that I'm bagging on them, but I do not prescribe to the mentality that the big corporate overlords are bad guys. I think that is oversimplistic. I think it's silly. I think that, profit incentives can make people make bad choices in any industry, anytime. But the people that do the research at these companies are good people doing it for the right reasons. So can companies with huge bureaucracies and multiple different people trying to multiple chefs in the kitchen trying to make choices, make bad choices? Absolutely. But I don't want anybody to think that I'm saying that there's like an actual corrupt intent or anything like that on part of any of these people. I just think sometimes it's easy to lose the forest for the trees. And I think that the case study of optic neuritis as a microcosm of MS to get clinical approvals is absolutely right now deeply lost in the forest, and people just can't see it. And I don't know why. It seems so obvious.
Well, and I think there's lots of reasons that cause that pressure, right? I mean, there are reasons that could be financial. There are reasons that could be, job security. You need to publish to be able to continue to get funding. There is pressure just in terms of, we want to help people now. So, do we want to take 10 years to develop this if we could maybe shorten that time frame? But as you say,
and that's 100% true. And I will say that you also there's more impact to saying you've done something for MS than optic neuritis. There's no doubt about that. You'll get more press, you'll get a higher impact journal, all those things. And there's obviously drug protections are only so long. So if you get an approval for optic neuritis, but you just burned 8 years on your patents before you even start looking at MS, that could really be, negative, a hindrance to your bottom line. There's IP strategies you can use to get around that, but that doesn't take away the risk that you'll diminish your patent protection by doing one, then the other, as opposed to doing both at the same time. But that being said, I don't find that to be a compelling reason to risk it failing wholesale. Right. That's the part that I struggle with. But Ari Green's lab, I mean, they've done some really good work. They've done a lot of really good kind of revelatory things. Clemistine and optic neuritis is just one good example. It is really surprising that other people kind of haven't taken. I mean, honestly, from the basic science perspective, I think it's odd to me, because I think from the basic science perspective, it's EAE has become so prevalent and easy at the basic science level that an optic neuritis model is a little bit more obscure and a little bit less commonplace. That on the basic science side, you've almost biased yourself towards the MS system. So that might be part of the bottleneck too, is that the preclinical animal models for optic neuritis aren't as commonplace or as straightforward as just immunizing animals and inducing AE. So that might be a role as well that perhaps I haven't thought enough about.
Yeah. Well, look, we could. I know us, and I know how we are on calls. I know how we are when we were in Sweden and in New Zealand together. We could talk like this for hours. If you have stayed until the end of this video, congratulations, because this has been a long chat.
You get a gold star.
We want to know, we want as much feedback as possible on this. We want to know how you enjoyed these style of chats, whether you need us to keep them shorter, whether you're happy to watch something this long. We're definitely going to be looking at doing some live broadcasts so that you can interact directly with both Travis and I, and ask us questions as we go through. But tell us what you thought. Hopefully you've enjoyed this chat talking about a topic that we know is really interesting and important to our community. Thank you very much for joining us for this, Travis, and sharing your expertise in the field. Sure. It's much appreciated from us, and I know it will be from our community. And I look forward to doing many more of these with you in the future.
Me as well. Hopefully we'll get to do some in person sometime soon also.
That would be great. All right. Thanks.
Great. Thanks a lot.